So how do these two cells, blah, blah, blah.
It's not so easy.
The final
it's
product
just
is
easy immunology.
Final product is easy. The way is not easy.
No.
The way is filled with blood, sweat, and tears. Yeah.
Welcome to Easy Immunology. My name is Alex.
And my name is Stefan, and today we want to explain to you how the immune system detects pathogens.
To detect and afterwards fighting the pathogens, the immune system first needs to know what is self, so what is own tissue and own immune cells, for example, and what is non-self, like foreign cells or cancer cells or also pathogens. And for pathogens, we have, for example, viruses, bacteria, or also parasites. And the most important cells, maybe not the most important to defeat these pathogens, but the most important to censor and monitor these pathogens are macrophages and dendritic cells. Both have the same precursor, a monocyte, and the monocyte then can either differentiate into a macrophage or a dendritic cell. The pathogens are recognized by their specific structures. For example, bacteria have bacterial components which we don't have in our own body. And these components are called pathogen-associated molecular patterns, or short PAMPs. Next to the cell wall, it could be also the bacterial DNA, or for viruses also RNA. And these components are recognized by pattern recognition receptors, PRRs.
So if a immune cell, for example, like our dendritic cell or macrophages, recognizes these pathogen-associated molecular patterns via their pattern recognition receptors, they know that a foreign organism that is potentially dangerous and could cause a disease is around, and therefore they become activated. And what I would like to explain you now is the different types of pattern recognition receptors, and the most well-known and most well-studied are called the Toll-like receptors. Toll is a German word, actually, because when these receptors were discovered, the lab head was a German lady, and when she looked through the microscope and saw the effect of these receptors missing in Drosophila flies, she said, "Oh, this is Toll," meaning this is great. And these Toll-like receptors can be distinguished into two different classes, if you want to call it like this. The ones that are outside the cell, they are called like Toll-like receptor 5, which recognizes flagellin, which is the component of the bacterial flagellum, or Toll-like receptor 4, which recognizes lipopolysaccharide, which is a component of the outer shell of some bacteria, or some Toll-like receptors that recognize lipopeptides. And these receptors are on the outside of the cell because they recognize structures that are also on the outside of the pathogen. But certain pathogens can also invade our cells, and then they try to multiply within our cells, and they can be recognized also by other Toll-like receptors, like Toll-like receptor 3, 7, and 9. And they are located in the endosome because if certain pathogens infect our cells, they end up in this endosome. This is a special compartment in the cell where stuff ends up that is taken up from the outside and then gets destroyed in there. And these receptors recognize bacterial or viral nucleic acid. For example, double-stranded RNA is something that doesn't exist in our cells, but it exists in the genome and in the replication cycle of certain viruses.
Next to the Toll-like receptors, we have also some other receptors on the surface of the cell. For example, the C-type lectin receptors. They are recognizing, for example, fungi and other microbial polysaccharides. But there are also receptors in the cytosol. When you imagine a cell, it's filled with liquid, and this liquid is called cytosol, and inside there is a lot of stuff. And for example, also the knot-like receptors, which can detect bacterial cell wall lipids and form a so-called inflammasome, which activates the immune cell really strong and also lead to the secretion of inflammatory cytokines, which are small signal molecules to also inform other immune cells that there's something dangerous going on. Next to the knot-like receptors, we have also the rig-like receptors. They can detect viral RNA. And there's also another form, the microbial DNA sensors. They are called CDS, cytosolic DNA sensors. And they are, for example, important to detect Ebola viruses.
Yes. And if we come back to our cells from the beginning, these dendritic cells and macrophages now express a lot of these pathogen recognition receptors, both on their surface and in their inside. And they can then, once they detect the pathogen, start an immune response, meaning alert other immune cells to the site of infection via these cytokines, or also go to the lymph nodes and then activate T cells. And this is something that we want to look at in the next video.
With this, we want to thank you for watching our video. We hope you enjoyed it and learned something, and we hope also that we see each other again. Bye.
Bye.
With this, I think we finished this episode when Stefan is clicking.
I clicked. Nothing's happening.